introduce Ms. Karen Rhodes. She did the EFT, and for half the population here that did not get to see it, we'd like for you to experience it.
Good morning. So I'm just going to do a round of EFT for those of you who are not familiar with it. EFT stands for Emotional Freedom Technique. And we did a round yesterday just to help energize everybody. So what we'll do is tap on some acupuncture points. And I'm just going to go ahead and say words. And if you can just follow along with me. Is this
I'll show that a bit today. That's program. Emotional and spiritual. So whatever, however you do that, but it has to be done.
Okay, so we're just going to tap on each of the points. We'll just tap on each of the points. Just say the words that I say. If the words don't resonate exactly with you, you can change them if you like. But I think people are maybe feeling jet lagged, a little tired, taking in all this information. So we're just going to do a little technique to help process all of that, to give us a little wake up, okay? So we're going to tap on this point, the karate chop point, and just say, just repeat after me, even though I'm feeling really tired.
Even though I'm feeling
I would like you to say it because you need to breathe while you're doing this, so we really have to inhale and exhale. Even though I'm feeling really tired, I choose to be energized. Even though I'm feeling really tired, taking in all this information, I choose to allow my brain to process it. Even though I'm feeling really tired, maybe jet lagged, I didn't sleep well, my heavy breakfast, I choose to be excited and energized. Top of the head, I'm feeling very tired. Inner corner of the eyebrow, so tired. Side of the eye, I didn't sleep much last night. Under the eye, I think I ate too much. Sugar. Under the nose, this is so much information, my brain feels very heavy. On the chin, feeling very tired. And then I'm just going to have you open your hands, go under the collarbone, just tap like this on both sides. I'm feeling very sleepy and lethargic. Under the arm, feeling very tired. I didn't sleep well. So much information, my brain's on fire. How exciting though. Feeling very lethargic. I feel like I need more caffeine. I feel like I need more sugar. Okay, now we're going to go between the ring finger and little finger. There's that little web space. Just rub on that, thinking about feeling a little lethargic and tired. You're going to close your eyes, open your eyes. Without moving your head, I want you to go down a diagonal, look in one direction, come back to midline, go in the other direction, come back to midline. Big circle with your eyes in one direction, eyes in the other direction. Hum a few seconds of happy birthday. Count to 9 fast. 1, 2, 3, 4, 5, 6, 7, 8, 9. Hum again. I'm so tired. I feel like I just need some more caffeine. We've got a long day ahead of us. We have lots of information. I'm really excited about learning all these things. I choose to be alert and awake. My brain can process information easily. I can apply the things that I learned. But I really am tired. I choose to be alert. I choose to be energized. My brain can take in all this information. I can process all this information. I'm very excited about all the things I'm going to learn. I choose to be alert. I'm very excited about the day and learning all this new information. Take a deep breath.
Wonderful. Wasn't that exciting? Okay, just a little bit of a commercial. I wanted to make a little comment for Dr. Waldo's lecture. It was really wonderful. Perhaps in the future we can also do a collaborate study with him, with manuka honey, coconut oil, and olive oil, a little study that we have been doing in the Philippines. So for you guys that are interested, manuka honey is out at the Secretariat for a very inexpensive cost and certified with its analysis on the UMM and all the good times. Okay, with all the energy now in the room, may I introduce the next speaker. He is the Director of Applied Cell Medicine Department at Schleswig-Holstein University, Kiel University, Chief Medical Officer of Liver and Kidney Transplantation at Schleswig-Holstein University, Kiel University, winner of George Herber Award because of long-term tolerance induction of allogenic organ transplantation, received a Rudolf Pilchmeyer Award because of the use of the peptides and stem cells as an outstanding contribution to tolerance-induced immunity, award from the German Society of Surgery, appointed as a lifetime professor of biotechnology at the School of Biotechnology at the Royal Liberty University in London, member of the European Transplantation Society, member of American Association for the Advancement of Science, and director of the Department of Applied Cellular Medicine, member Delphi Group concerning embryonic stem cells for human use. Established by the German Ministry of Health and German Ministry for Education and Research. A guest professor, Faculty of Medicine at the UITM Sungai. Let us welcome Professor Dr. Fred Fandrich.
Thank you very much for this warm introduction. I'm happy to be here. Welcome, honorable guests and colleagues. Today, my task is to talk about cancer metabolism. Can I have the first slide, please? Well, when I got into this subject, as you heard, by profession I'm a surgeon. I did a lot of oncology and transplantation surgery, and in 2009 I got the offer from my university to chair a department for cellular medicine. And I will give you in my talk tomorrow morning much more details on how to use your own cells in order to modify the immune system. But today, the committee who invited me picked the topic of cancer metabolism. So I honestly tell you, I had to study because cancer metabolism is a very complex subject, but I think it's a very important subject because if we understand the origin of cancer, what really drives cells to turn into a cancer cell, into a cancer stem cell, we have a much better target to treat cancer, and that's important. Well, I like this proverb from this Nobel laureate who says, discovery consists of seeing what everybody has seen and thinking what nobody has thought. And I always tell my students, I think that nature itself gives us the most simple ways to solve problems. And if you look at nature carefully enough, you get a lot of ideas on how to treat our patients. Well, it was Richard Nixon, you know, the former president of USA, who in 1971 signed the National Cancer Act into law, declaring, I hope in the years ahead we will look back on this action today as the most significant action taken during my administration. Well, as a matter of fact, by then, US spent billions of dollars into the research of cancer, cancer metabolism, and potential treatments. But if you look at what came out of all this huge money budget invested into cancer research, and this is a survey from 2019 from the American Cancer Society. You see that the estimated new cases and estimated deaths from various cancers has not changed very much since the years 1971, which is illustrated here for men. Of course, lung cancer really has been reduced over the years. Here's 1971, but for instance, pancreas, liver cancers, and prostate are again increasing. And the prediction is that within the next 10 years in US, more people will die from cancer than from cardiovascular diseases. And the same how it's true for women cancer. Even there are some achievements. The picture is pretty much the same, and pancreatic, liver, and uterus cancer are increasing. So we have to confess that the war on cancer by now has not been won or solved. So what is it what we can really do? And I'm not going to give you any treatment regimens or strategies right now. This lecture is in order to understand what are the implications genetically and metabolically which drive cancer development. And I hope my colleague later on my colleagues are giving you some examples how you can address this. I will give you some ideas, but this lecture is more to understand what's going on. Now, in the academic field, there has been, and there's still ongoing, a big discussion whether the trigger of cancer formation is a mutation of genes or the trigger is a change of metabolism in the cell, which secondarily changes the genes. Now, this is quite difficult to answer, and I got aware of this conflict, and I will give you pro and con for both. I'm not really at the point that I can say it's the one or the other, but maybe after my lecture you will understand. So, one of the most cited and famous papers in Cell was about the hallmarks of cancer, written by Hanahan and Weinberg. And actually, when they started describing the most important hallmarks of cancer, the original publication had six topics. Then, in 2011, they extended that and added the metabolism, the reprogramming of the metabolism of cells as one of the major hallmarks. It is clear that the sustained proliferating signaling is one of them, evading growth suppressors, and tumor microenvironment, but also, and this is very important, the metabolism was added into this scheme in order to explain how cancer cells can attain the proliferative potential and how they can evade chemotherapeutic treatment, how they can evade immunotherapy. Well, one of the proponents of metabolism was a very famous person in Germany. His name is Otto Warburg. This is Otto Warburg. Professor Warburg worked as a biochemist at the Charité University in Berlin. And he was the first to identify that actually what you can see in all cancer cells, what is happening is a change of the mitochondrial respiration. turning into glycolysis, aerobic glycolysis. And I'm going to explain what that is. One thing which attracted me very much and came to my attention was this publication from the 90s, which made a comparison between the tumor environment of healthy cells and tumor cells. And what it actually shows is quite simple. Here you have different tumor entities: glioblastoma, astrocytoma, uterine cancer, melanoma. And given here the x-axis is the extracellular pH. And what you see is that if you measure the pH in the tumor environment of these different cancers, it's significantly more acid than if you compare the tissue from the same patient in the healthy environment. So apparently the acidity in the tumor environment is one as a major problem which allows the tumor to grow. And it's important to understand why this is happening. Well, if you want to go into the field of cancer metabolism, it is absolutely must to read this book from Fandrich, an American biochemist and geneticist. He has studied genetics and biochemistry. And in his book, he lines out the implications of the mitochondrial metabolism for cancer formation and how it is related to oncogene activation and all the other implications. And I want to give you a little bit an insight on what's going on. This is this book, and up here, this shows a presentation of deletions and DNA abnormalities from 24 different pancreatic cancers. And what you see is that each cancer on its own has quite different amount of mutations, chromosomal defects, and that shows that our endeavor to treat cancers with one type of targeting a certain mutation or a certain metabolism in the cell can never be successful, because even within the same tumor cell, you find a lot of heterogenic pathways activated. And the same is true for this picture here, which shows, actually it's not so clear, can you see that? No. What it shows are different activated suppressor genes, mutated suppressor genes, or activated oncogenes, which are apparently driving glioblastoma growth. And what you have to learn here at this point is that in each of these cancers, even you have a loss of function of suppressor genes which control the growth and proliferation, and at the same time you have an activation of oncogenes. However, and this is what is illustrated here, even in the same tumor from one patient, you find cells which show quite different types of suppressor gene downregulation and oncogene upregulation. So, and one thing on top of that is that 15% of the cancer cells, roundabout, in cancers do not show any of these mutations. So they are not affected at all genetically. So by targeting certain mutations, by small molecules, antibodies, and so on, you will not achieve to reach the whole mass of tumors. And that has important implications if you treat patients' tumors with a certain regimen which targets certain mutations. You will miss certain cells within the tumor mass, and that will help them to become more and more resilient. And that's another topic of chemoresistance, which is directly related to the metabolism and the treatment, which I want to line out later on. Now, one of the intriguing arguments in favor of metabolism as the starting point for cancer development are these experiments which have been conducted accordingly to what I'm telling you now. Here you see a normal cell with a normal nucleus, normal cytoplasm. This is considered a tumor cell. You see the shape is different, the cytoplasm content is different, the mitochondria altered, and the nucleus shape is different. Now, what people did is they bring the nucleus of a tumor cell, they transfer that into the cytoplasm of a normal healthy cell. And what they observed was that in most cases, actually more than 90%, the cell stays healthy. Which means that the genetic changes in the nucleus were not able to convert the cancer status into the healthy cell. However, if you bring a healthy nucleus from a healthy cell and transfer it into the cytoplasm of a tumor cell, the cell turns into a cancer cell. Which means that the cytoplasm and the related metabolism were able to program the nucleus in a way to turn into a cancer cell. And keeping that in mind, it underlines how important metabolism really is in the context of tumor formation. Now, I mentioned already Professor Seifried, the biochemist, and he really has lined out the importance of the reprogrammed metabolism in tumor cells, and has pointed out that here, with essentially all hallmarks of cancer, including aerobic glycosis, can be linked to impaired mitochondrial function and energy metabolism. A view of cancer as primarily a metabolic disease will impact approaches to cancer management. And he gives a lot of examples in his book. However, it is not that easy. You cannot just claim that metabolism is everything, which I will show you. What is important, and this is part of my lecture to you to understand, is to explain and to differentiate the different types of metabolism tumor cells use to circumvent the treatment approaches like chemotherapy, and to create an environment where they can contain their own proliferation and growth. And we have to differentiate between oxidative phosphorylation, and I will tell you in the next slides what this means, versus anaerobic glycolysis versus aerobic glycolysis, and versus substrate phosphorylation. I will try to explain what this means. So here is the cell, and the cells have to produce energy, and tumor cells have to produce even more energies because they are fast expanding and growing. Now, what's happening is that for energy, the cells use basically proteins, sugars, and fats, that's clear. And the breakdown, glycolysis of glucose is happening in the cytoplasm. The same happens for fatty acids, and then converted metabolites are introduced into the mitochondria, where normally by use of oxygen and cellular respiration, these compounds are used to produce ATP. ATP is the driving fuel of every, or more than 90% of enzymatic reactions in our body. And you will hear about mitochondrial physiology and biology from Professor Aminuddin later on, but I have to give you some ideas on what mitochondria are for. ATP, as I say, is the major driving energy fuel for our body, and if you weigh 80 kilograms, you produce 80 kilograms of ATP per day. Imagine. So, like a heart, a muscle cell, or a neuron cell contain between 10 and 20,000 mitochondria per cell, and they produce billions of ATPs every second. And this is amazing how this is functioning. But you can imagine if this mechanism is disrupted in some way or the other, then free electrons are released from these mitochondria, and these radicals, reactive oxygen and nitrogen species, they can cause a lot of damage. They cause inflammation, they cause genetic instability, and they cause chronic disease. So the close correlation between metabolism and disease formation is, to one extent, very, very much associated with the physiologic activity in mitochondria. Now, tumor cells, in a lot of cases, they stop cellular respiration and change to other forms to create energy. And that's so interesting, and I want to go into that now. But before I do that, I have to explain aerobic versus anaerobic glycolysis. Now, the breakdown of glucose to pyruvate has 10 steps, and basically this breakdown is the first part which is happening outside the mitochondria to produce either pyruvate or lactate. In situations where you have oxygen, the breakdown is into pyruvate, and then pyruvate is broken down into acetyl-CoA, which enters the citric cycle, which I will show later. If there is not oxygen, like you're exercising, you're straining your muscles, and you cannot keep up with the oxygen consumption, then oxygen is missing, then the cell can turn into lactate formation. And this has implications on the amount of ATP which is produced. So, just for definition, the conversion of glucose to lactate under anaerobic condition is called anaerobic glycolysis. The conversion of glucose to lactate in the presence of oxygen we call aerobic glycolysis, or Warburg effect. And this is exactly what's happening in cancer cells. Even in the presence of oxygen, for certain reasons, they change from cellular respiration into lactate formation. Can you go on? It's not moving yet. Okay. So now let me explain what is phosphorylation. The addition of a phosphate to a molecule or protein is called phosphorylation. Enzymes that phosphorylate molecules are called kinases, and typically kinases use phosphate from ATP to add to a molecule. like shown here. The first step in glycolysis is the formation of glucose 6-phosphate, and this is performed by an enzyme called hexokinase, and it uses ATP and takes the phosphate molecule and attaches it to the carbonate group on the glucose, and you have glucose 6-phosphate. So this is phosphorylation. However, in substrate level phosphorylation, this means that the synthesis of ATP by the direct transfer of phosphate of the phosphate group from the substrate. Here's the substrate, D-phosphate, glycerate. The phosphate is detached and added to the ATP in order to produce ATP. And I told you ATP is the crucial energy which we need and tumor cells need. So what tumor cells do, they change away from the respiratory cellular energy ATP production away to substrate level phosphorylation and produce ATP in a different way. Now, to summarize, oxidative phosphorylation, which happens in the mitochondria, is the use of O2 oxygen to oxidize electron carriers like NADH or SADH in order to generate ATP. I'll show you in a second how this is done. ATP is made through a mechanical reaction. It's not an enzymatic reaction, and this occurs in electron transport chain inside the mitochondria. This is the normal way how cells produce ATP and energy. Just to show this in detail, glucose is taken, you put in 2 ATPs, glycolysis starts, and at the end you get 2 molecules of pyruvate. This is a 6-carbon molecule, these are 2 molecules of 3 carbons. And by doing that, you generate 4 ATPs and 2 NADH, which are the electron carriers important for the ATP production in the mitochondria. Now, the next step, you take the 2 pyruvates, and there's an enzyme called pyruvate dehydrogenase complex. This enzyme breaks down pyruvate 3-carbon molecule into acetyl CoA, which is a 2-carbon molecule, and 2-acetyl CoA then enters the mitochondria, and inside the mitochondria it delivers the NADH as electron carrier. We'll go through that in a second. And within the mitochondrial matrix, there is the 3-carboxylic acid cycle or citric acid cycle or Krebs cycle, and that takes a bunch of enzymatic reactions to take the 2 acetyl CoAs and to convert them in 6 molecules of NADH, 2 molecules of FADH, produce CO2 and 2 ATPs. So then, after that, or just to reverse, just to remind you, if oxygen is lacking, then pyruvate is not degraded, converted into acetyl, but is converted into lactate. And this is happening in the cytoplasm, whereas the degradation of acetyl CoA and the production of these electron carriers are all happening within the mitochondria. Here you see a mitochondria. You will get more details on that. It has this cristae, it has an outer membrane, and it has an inner membrane. And the space between the inner membrane and the outer membrane, this is the space where all the protons are accumulating, which is used during the electron transport chain activation. And this is important, as I will show you. Now, the point I want to do here is to make here is that if you convert pyruvate into lactate, you just have a gain of 2 ATPs per molecule glucose, where you have a much higher gain of ATP production if you undergo the citric acid cycle and then the respiratory oxidation in the mitochondria. So, the normal energy production of ATP undergoes glycolysis, pyruvate oxidation, citric acid cycle, and then oxidative phosphorylation in the mitochondria. And this is the electron transport chain, where the electron characters they are oxidized to NAD+, and while they deliver one electron, one proton is pumped into the inner membrane space of the mitochondria. And this is happening in complex 1 and complex 3. Complex 2 is delivering electrons from FADH, 1 from NADH. Now, the electron transfer is permitted with Q10, ubiquinol Q10. Coenzyme Q10 is the electron carrier between complex 1 and 2, which is on complex 3, and then cytochrome C carries on the electrons on complex 4, where oxygen is used to oxidize the oxidized and produce water. Important is, and this is one thing you need to know, if you lack coenzyme Q10, and the more you age, the lower you can find your coenzyme Q10 levels. The electron transport chain is also hampered in the mitochondria. And that leads to oxidative stress. And oxidative stress will lead to inflammation and all the complications from that. So one of the major things you have to do with the patients, especially in nowadays, you have to supply enough Q10 to keep the mitochondria running. Professor Aminu will show you a lot of examples how chronic diseases are related to mitochondrial dysfunction, and that basically is related to lack of Q10. But there are a lot of other compounds which are important. What's happening here now, to come back to the respiratory energy production, you have basically 3 different gradients. You have a gradient between high level of protons in the inner membrane versus lower proton in the matrix, and you have low pH up here, higher pH inside the matrix, and there is a membrane potential because of the different charges, electron charges inside and outside. And all 3 are important to drive this ATP synthase, which will allow to take one proton from the inner membrane space to pump down, and by pumping down, the whole molecule is turning like a turbine and produces ATP by putting together an organic phosphate molecule with ADP. And this process of mechanically putting together ADP and phosphate is called chemiosmosis. And all this is part of the electron transport chain, and the whole process of donating electrons and producing ATP by chemiosmosis is called oxidative phosphorylation. And this is the normal mechanism to produce ATP. Everyone is using now, sitting here, breathing oxygen. Now, why? Oh, perhaps one more thing. 30% of the ATP we produce comes from glucose, and 70% comes from beta oxidation, from fatty acids. Because one round of oxidative phosphorylation from a glucose molecule will produce approximately 36 ATPs. If you take fatty acids, it will produce 129 ATPs. So fatty beta oxidation produces much more ATPs than glucose breakdown. That's important to know. Well, and inside the inner membrane, here this is just symbolizing the ATP synthase, which generates these, pumps down these protons to produce ATP. And there are millions of these channels inside the mitochondria, and permanently this whole thing is happening. So it shows to you how fragile the system is. If you have an abundance of H plus protons in this pool up here, and you do not exercise and you overeat yourself, then more and more protons are accumulating here and cannot be used. And that will automatically instigate a process where electrons are lost and free electrons are spinning around and damaging the mitochondria. And then you need to know that the mitochondrial DNA, which is its own DNA apart from the nucleus, is not covered and sheeted by histones. So it's much more fragile for damage. And that is why there's a much higher risk to damage mitochondrial DNA from free radicals than primarily the nucleus DNA. And that is why if the metabolism is disturbed and more ROS is formed, there's a higher probability to first have damage inside the mitochondria. Well, having said all that, I can go one back. The question is, the real critical question is, if the mitochondrial respiration is the optimal way to produce ATP, why do most cancer cells reprogram metabolism and turn back to aerobic glycolysis and produce lactate? Well, there are reasons for that. Here is from this book from Fandrich and his publications. If you look at the percent of ATP production in a cell, if the cell is normal, it produces by oxidative phosphorylation. I just explained that, cellular respiration. In tumor cells, you have, with tumor progression, an ongoing conversion from oxidative phosphorylation to substrate level phosphorylation. And this is really a common denominator which is true for all tumor cells. So why is this happening? This is happening because the cell wants to shut down certain genes which protect cells from running into uncontrolled proliferation. And by changing the metabolism inside the mitochondria, a lot of different genes, and I will explain the pattern of activation to you in a few seconds, a lot of different genes are activated inside the nucleus because there is something called retrograde response, where information from the mitochondria is passed on into the nucleus and vice versa. So there is an ongoing communication between mitochondria and the nucleus, which determines which genes are activated or downregulated, and these genes, they determine whether you have growth signals, whether you have angiogenesis, and all the detrimental factors going on in cancer cells. Now, the proponents of the metabolic theory of cancer development, they claim that the first instance of damage by virus, by inflammation, by hypoxia, by radiation, by carcinogens is hitting the mitochondria. And that damaged mitochondrial DNA, that changes the metabolism, that impacts on the genome instability within the nucleus, and all the consequences. Well, that might be true, but there are other people in the scientific academy which have a different view on that, which I want to explain. Well, I think I need to show you this before I go ahead. Yes. Or maybe let me just go ahead a little bit. Yeah. So the other view, the contra view, claims that first you have mutations of very important genes inside the nucleus. They impact on abnormal metabolic phenotype, which includes mitochondrial disorders. That changes the tumor environment, and the tumor environment strikes back and in a vicious cycle and increases the damage to the mitochondria, and that has all these consequences to allow tumors to grow. And this is only a small spectrum of important suppressor genes and oncogenes which determine metabolism inside the cells. Now, you see here in a normal healthy cell, you have a glucose receptor, GLUT1 or GLUT4. You have the normal glycolysis, and then you have pyruvate dehydrogenase that forms acetyl-CoA, and the normal TCA and respirate. Now this can be blocked in rapid phase by certain suppressor and oncogenes which are activated. But before I go into that, I have to go back now. I didn't show you this. I want to just to show you one example. One example which is very important, and you have to keep this in mind when you treat your cancer patients. Virus contamination can affect cells, and the virus can change the metabolism inside the mitochondria. And that has detrimental consequences. And I want to give you an example of the cytomegalovirus. In this publication in Nature from 2019, what they say down here is, we and other laboratories have shown that PP65, which is the CMV antigen, is expressed in more than 90% of glioblastoma specimens, but not in surrounding normal brain cells. Which means that, and that was shown also in this publication, as long as you do not treat the virus inside the cancer stem cells, it will prevail and it will protect the cells from chemotherapy and from immunotherapy. So what they did actually was they produced dendritic cells, and tomorrow in my lecture on immunotherapy I will give you much more information on how to do that. Dendritic cells are monocyte-derived cells which you can load or pulse with certain antigens, and then they express these antigens and they elicit a specific T cell response against the antigen which is expressed. Now what they did here, they used tetanus toxoid to recall the immune system, to activate the immune system very unspecifically, and they pulsed the dendritic cells with PP65. And PP65 is a specific antigen expressed on CMV virus. So injecting these dendritic cells back to the patient, they could elicit an immune response against CMV. And that's so intriguing here. Where is this picture? Sorry. Yeah. This dendritic cell vaccination you see here allowed 50% of the patients with glioblastoma grade 4 to live more than 5 years. This is extreme. The normal survival of a glioblastoma patient is 14 point something months, including radiation and temodal treatment. And with that, they have 50% 5-year survival, and after 5 years, you expect patients to be cured. So it just demonstrates how important it is to, at the same time, to treat virus disease if it is evidently affecting the tumor. Okay. So to summarize this first part, any unspecific condition that damages cells, oxidative correlation, but is not severe enough to induce apoptosis in the mitochondria, in the cells, can potentially initiate the path to malignant cancer. So inflammation, radiation, intermittent hypoxia, germline mutations, viral infections, and the tumor microenvironment have been shown to instigate mitochondrial problems, metabolic reprogramming, and cancer formation. So I'm coming back to that. The other view, which holds up that first there's genetic change and then there's reprogramming of metabolism. So I explained the healthy cells, but here what happens if one of these suppressor genes or oncogenes are modulated? Well, P13K is a protein which activates a pathway which secondarily downstream activates another transcription factor, mTOR, and that's a key metabolic integration point, mTOR. This is where metformin actually is active, protecting against cancer formation by regulating mTOR. And mTOR actually activates HIF, hypoxia-inducing factor, and that has considerable impact on the metabolism inside the mitochondria and for the cancer cells. I now explain what HIF is doing. Under normoxic, meaning normal oxygen levels, HIF subunits undergo oxygen-dependent hydroxylation, meaning HIF cannot be transcribed in the nucleus and is inactive. However, under hypoxic conditions, HIF is responsible for gene expression changes during the cellular response, and it amplifies genes encoding glucose transporters. GLUT1, GLUT4 are upregulated, and glycolytic enzymes. It activates pyruvate dehydrogenase kinases, which inactivate mitochondrial pyruvate dehydrogenase complex, and thereby they reduce pyruvate influx into the citric acid cycle and decrease oxidative phosphorylation. So, what does that mean practically? You have a tumor, and the tumor is growing, and normally tumors grow faster. 10 minutes left? Okay. Well, I was taken 10 minutes away when I started. Okay. You have a hypoxic condition in the center of each growing tumor, because in the beginning angiogenesis is slow and oxygen cannot be supplied to growing tumors. And that automatically on its own induces hypoxia. And that hypoxic environment, that will allow all these complicating activations of oncogenes, which then on their own drive cancer formation. I want to, not to go to all this, what I want to show you, if I have only 10 minutes left, one thing is that these genes is especially another oncogene which is activated by HIF, is MYC. And MYC activates pyruvate kinase, which slows down the glycolysis from glucose to pyruvate. And by slowing down this last step, it allows other pathways to activate and to produce NADPH as electron carrier and macromolecules for the building blocks necessary to proliferate in more cells. And so PK, the pyruvate kinase, which slows down glycolysis, provides an advantage to cancer cells because by slowing this down, it allows carbohydrate metabolites to enter subsidiary pathways to produce building blocks cells need to proliferate. And other pathways, like the pentose phosphate pathway, TPP, is activated. And this is the way how tumor cells circumvent cellular respiration by, at the same time, providing molecules which fuel the building blocks for growth and proliferation. Now, the one much more important thing which links both together is this publication which shows that actually tumor cells are so intelligent, they inform neighbor cells like fibroblasts to acquire cancer properties which will function like a nutrient source to fuel the cancer cells. What does that mean? It has been shown, and this is also in this publication, that a tumor consists of very, very different types of, as I mentioned before, cells with different mutations and different metabolism. And some of these cells inside, they have quite a high stemness. They're very similar to self-renewing stem cells. Others are dormant, they don't have that. But, and this is the important point, and this is pointed out here, with the multi-modality therapy concept which comprises surgery, radiation, chemotherapy, what you normally achieve is you reduce 90% or even more of the tumor mass, but you have some minimal residual disease of cells which, after the first initial treatment, convert metabolism and turn more and more into stem cell-like cancer cells. And then, if the cancer relapses, you have more and more aggressive cells in the tumor heterogeneity, and it gets more and more difficult to defeat these cells. And how are tumor cells doing this? If you compare the metabolic status in these various phases of tumor development, you see in the beginning you have quite a lot of aerobic glycolysis and other pathways, like tumors use glutamine a lot to provide the purines and all the building blocks. And the more the tumor gets aggressive, the more it changes the metabolite and the reprogramming inside the tumor, and the more it produces lactate and produces acidity. And this acidity is important. Now, it has been shown, for instance, for the bladder cancer, if you treat bladder cancer with chemotherapy, what happens? Here is a sleeping, a dormant cancer cell, by treating this tumor formation with chemotherapy, these cells are killed and they release a prostaglandin E2, and prostaglandin E2 can activate dormant cancer cells to start to get aggressive and to proliferate. So by killing certain cells inside the tumor mass, you can produce compounds which are released from the dying cells, which will then activate and make the tumor more aggressive. This is a matter of fact. So this you have to keep in mind. And the second thing is that cancer cells create an environment which is sort of hypoxic and contains a lot of free electrons which can change the metabolic status of cancer-associated fibroblasts. And fibroblasts then turn into the Warburg mechanism, the aerobic glycolysis, and they feed again the cancer cells. So what you see is that the fiber-associated cancer cells, they take up glucose and convert to lactate and reverse the lactate back to the tumor cells by certain monocarboxylate transporters. And inside the tumor cells, all of a sudden, you have a reverse Warburg, a reverse aerobic glycolysis, where again the tumor cells converts lactate to pyruvate and undergoes normal respiration. Which means, at the beginning, you have aerobic glycolysis to produce lactate. The tumor produces an acid environment, ROS environment. That changes fibroblasts and other cells in the environment to become a unit of energy production of lactate for the tumor cell itself. So you have two different types of metabolism going on in the cancer cell and the cancer-associated cells, which is called metabolic symbiosis. And this is also an option to block this channel, the monocarboxylate transporters. There are now medications tested which block this in order to stop these cells from the nutrition of the associated fibroblasts. But it shows to you how complex the situation is. It is not only to treat the cancer cell itself in the tumor environment. You need to change the tumor environment itself before you can be really effective. And that is what I want to show here. Now, tomorrow I will talk a little bit about cellular immune therapy for tumors. But, and this is the normal thing what happens. You have a tumor, and normally the immune surveillance by your immune system activates NK cells. NK cells can destroy tumor cells, and the antigens from the destroyed tumor cells can be presented to dendritic cells. Dendritic cells pick it up, and then they digest it and present tumor antigen on the cell membrane. And then they go to the lymph node, and in the lymph node, dendritic cells present the tumor antigen to T cells, activate cytotoxic T cells, and the T cells circulate back and look for the tumor, and where they see the tumor antigen, they attack and kill the tumor. However, what happens is that the tumor cells are smart, and they produce an environment where they activate other immune cells to suppress the T cells. And this environment, and this is important here, I go over this publication. You can look this up. There's lots of publications showing that. This environment will activate genes which activate HIF and lower the oxygen, and then you have an acid environment. And in an acid environment around the tumor, chemotherapeutic regimens cannot access the tumor cells anymore. And important, activated cytotoxic T cells cannot infiltrate tumors professionally to kill tumors if the environment is acid. And as I showed you in one of my first slides, that is why you see in tumors a much lower extracellular pH than outside. Now, here are all the different impacts on acid environment, on NK cells, on dendritic cells, on cytotoxic T cells. They lose their power to kill tumor cells in a low pH environment. So, I will be finish in a second. The cells have sensing, pH sensing molecules in the membrane, which when they sense acidity, they also change the glycolysis into aerobic lactate formation. And they also activate oncogenic pathways to let the tumor cell grow if they sense acidity outside the tumor. So what you have to do actually, you have, I'm not going into that, you have to, just because I have to stop now, you have to treat that. Now, as a matter of fact, you know, conventional treatment, surgery, chemotherapy, radiation, a cure of that in cancer, less than 5%. Because the tumor environment will support immune escape and reproductive metabolism. Now what you can do is you can pH balance the environment, you can induce and boost mitochondrial function and reinforce OXPHOS, the respiratory energy. And oh, can you go on one, please? Next one. There's one missing. My pointer doesn't work. And we have three compounds, and that's the last thing I want to tell you. We have three compounds which we use complementary to chemotherapy or other ways on how you treat tumors, which are very important. And with these three compounds, I will explain in a second, you can reduce the toxicity from chemotherapy and you can increase the efficacy at the same time and increase immunity to kill cancer cells. Now, this compound is called Relief. It's based on procaine, but the former procaine was bound to hydrochloride acid. And what we did, we detached the hydrochloride and we embed the procaine molecule into hydrocarbonate. And if you infuse this Relief procaine molecule into a patient, the high amount of positrons, hydrogen and saline, they will actually target the protons with the procaine into the tissue where you have high acidity. Because in high acidity you have high electron density. And then you can measure that in animals to balance the tissue pH within 30 minutes to normal pH. And that has a dramatic impact on the metabolism of the tumor cells. Dr. Longo pointed out confusing as a major point in his concept, and this confuses tumor cells. And at the same time, what we do simultaneously, it has been published that this procaine has cancer prevention efficacy. I don't want to go. We boost the mitochondria with a lot of important organic amino acids and enzymes and vitamins which are important to sustain normal electron chain transport activity. We combine it with NADH glutamine and coenzyme Q10. And last but not least, I'm finished in a second. Give me 1 minute. We have one compound which is extremely powerful to reverse the aerobic lactate formation. And this compound is called DCA. Now, what I showed you in any hypoxia, tumors activate hypoxic-inducible factor, and that blocks pyruvate dehydrogenase by activating the kinase. Now, what DCA does is reverses, blocks pyruvate dehydrogenase kinase, and thereby allows pyruvate to form, to go into, by acetyl-CoA, to go into the citric cycle, and to reverse the metabolism back to respiration. And thereby, apoptosis is allowed again in the tumor cell, which is blocked if the tumor produces lactate, and the growth rate stops. And there have been a lot of publications on that showing how DCA on its own can help in cancer situation. This is just a PET CT that shows the glucose going into glioblastoma tumor in comparison to non-glioblastoma tissue, and it has been tested, for instance, in animals. It works in sarcoma. You see, this is the control-treated animals. Here, the DCA-treated animals where the sarcoma is shrinking after 21 days. And there's huge other publications, lists, hundreds of publications which support this concept. And I want to summarize this at the end. Cancer metabolism. Cancer uses a variety of energy-producing pathways, which I have lined out. It creates a large spectrum of heterogeneous tumor cells within the same cancer. Multiple oncogenes are activated, multiple suppressor genes are mutated, inconsistent genomic signatures of tumor cells within the same tumor, which make it hard to address targeted therapy. Activate the stromal cells like fibroblasts, which promote tumor growth. And all this suppresses tumor-specific immunity and acidification of the tumor environment. Now, as my take-home message, intratumor heterogeneity due to the presence of cancer stem cells is primarily responsible for our inability to induce the same therapeutic effect among cancer cells as a whole. And minimal residual disease is expected to contribute prominently to the latent relapse and distant metastasis. Last but not least, we are far away from having won the war on cancer. Targeted therapy focusing entirely on negative patients will not improve outcomes. Immunotherapy alone will also fail. CAR T cells, infiltrating lymphocytes, checkpoint antibodies, oncolytic virus, all that can only work if the tumor environment is normalized. Same is true for chemotherapy treatment. So you have to restore the physiological tumor environment to a physiological environment and have preventive approaches, mitochondrial medicine, to increase chemotherapy efficacy, to reduce related toxicity, and to strengthen tumor-specific immunity. Okay, I'm done, and I want to finish with my friend Albert Einstein. If you can't explain it simply, you don't understand well enough. I hope you got some of the messages I explained.
Great. That's a lot of information. Wasn't it useful that we had that energy exercise earlier? And now I think we do need to feed ourselves with some ATP. Okay. For the question and answers, can we reserve that for tomorrow's lecture with Dr. Fred? Is that okay?
Yeah, that's a good idea to do that in combination with my other lecture.
Thank you. Okay. Meanwhile, but we will not do the token of appreciation tomorrow. This is going to take a few minutes. So can we invite back Dr. Hoover Lin and Dr. Mazia for the token of appreciation for Dr. Fred up on stage. Thank you.
Just 30 minutes. Just 30 minutes.
Thank you.
very good news for those who have bought any of the three books that we mentioned this morning. We have made a special arrangement on the book signings by the author. It will be today at 3:30 p.m. During the tea break. We'll be at the VIP holding room on our